Hello. Okay, thank you, Dr. Thiru, yeah, for your time, giving some your expertise here in our forum. But now we have the new name, the Indonesian Metabolomics Associations. Yeah, thank you very much. So, yeah, everyone, let's start our this lecture in the Metabolomics forums. So here we have Dr. Thiruventhan Karunakaran from University Sains Malaysia. But I think you can yeah, give some information about you later for the audience. Yeah. Uh, okay. Yeah, you can talk about 1 hour and maybe 30 minutes maximum for the question and answer. Yeah. So the Zoom is yours, Doctor. But so sorry, maybe I close my camera because I have a lecture this morning also. Yeah, but I
Okay.
still okay.
Okay. Thank you, Prof. Rafi. Thank you very
Yeah.
much for giving me the opportunity to present a topic. I think it's too early in Indonesia, yeah? 9 a.m. So in
Oh,
Malaysia,
no, 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 not only. Usually we have class also at 7.
oh, it's very early.
Yeah.
Okay. So shall I share my slide? Okay. I think everyone can see my slide. Okay. Thank you. So, yeah. Thank you very much to Prof. Rafi for giving me the opportunity to present a topic on the science of kratom, insight into its phytochemistry. So I know that a lot of you have been exposure to phytochemistry, natural products, yeah. So the name of kratom ataupun ketum, uh, have you heard about ketum or kratom? I think most of Indonesians also knows about this plant because recently we have a collaboration with the we coming out with the mini conference or mini meetup, scientific meetup in Bali, Indonesia, which is a co-joint organized by the BRIN. Yeah, BRIN is doing on ketum as well, kratom, and we have our partners from Thailand as well as from the Western country, US. We all are venturing into it, exploring, unraveling about the science of kratom itself. So today here, I'm giving a bit of information on the insights is phytochemistry because it is important when we talk about the plant science itself. Phytochemistry is a major part that we need to understand before we venture and go into the application of the plant itself. So my name is Dr. Thiruventhan from Center for Drug Research, University of Science Malaysia. So I think some of you might know University of Science Malaysia is situated in Penang. So I will give you a bit of introduction on that, and after that we venture into the we try to go into the topic itself. So this is a map of Malaysia. I think we are neighboring country. So a lot of Indonesians well known about Malaysia itself. And USM been situated in the state of Penang, or we known as Pulau Pinang. Yeah, it's a northern part of peninsula of Malaysia. So Malaysia is divided into two distinct geographical location. Yeah, one is the peninsula, one is the part of Borneo. Also neighbor with Indonesia, yeah. We have the state of Sabah and Sarawak, and here is the Peninsula Malaysia. So Kratom, ketum, it is well known to be found in the region of northern region, yeah, where it compromises in the state of Kedah, Perlis, as well as Penang, yeah, in some part of the other part of the state as well. But most heavily focused in this region, and USM having the advantage to be situated in the state of Penang, where we have the access to the kratom plantation itself. Within 2 or 3 hours, we can reach to the locations, and we able to collect, the leaf samples. Yeah. So the question here is, how does the phytochemical compositions, before we get into the title, the topic itself, is a question that we need to think about it. How does the phytochemical composition of Mitragyna speciosa, which is the botanical name of the plant, Kratom, influence its pharmacological activity and safety as a potential therapeutic agent? So a lot of people now trying to see whether we can develop Kratom to be a botanical drug. So we try to see whether it have the potential. But of course, recent research, this plant is a bit conflicted because he's heavily debated on his potential safety, toxicity, but also been discussed on the potential application, for becoming therapeutic agent, especially in analgesic, pain, as well for addiction treatment. So yes, Mitragyna speciosa, the classification of the plant belongs to the family of Rubiaceae. Rubiaceae, if you know, it is relative to coffee, coffee plants, Rubiaceae, and the genus is Mitragyna, species M speciosa. The common name, of course, Kratom or Biabia, and it's indigenous to this region, whereby is southern part of Thailand, northern Malaysia, and as well as a part of the Borneo Island. Also, it's been distributed to other part of Southeast Asia's region as well, but it's heavily from this part. So this is a picture was taken at the informal plantation in northern part of Malaysia. So you can see how the plant, Kratom, been planted. This is a location in the state of Kedah, So we can see how it been cultivated, planted in this region. So this is how the leaf looks like, the Kratom Mitragyna speciosa leaf. So the part that heavily used is the leaf, the leaf of Kratom. So we can see that how the leaf is look like, And then, of course, about Kratom itself. So historical claims on medicinal or pharmacological effects of chewing fresh leaf of Kratom tree, Kratom tree, which focuses on the leaf part itself, native to Southeast Asia, supplemented by the narrative accounts of potential health benefits of product derived from Kratom, and is consumed in the United States, US and other countries in the West, which has garnered an increasing interest among researchers, clinicians, and the public recent years. So it's a bit heavily debated plant, actually, So although there's a growing demand for Kratom-derived products, which is we focus is the processed leaf products, When we saying about Kratom-derived product, it is a processed leaf products. Among the United States consumers, there's also paucity of rigorously collected scientific data on human pharmacological safety profile of Kratom phytochemicals and very limited scientific data on their dose-related effects in humans. So there is some data is still limited for us to proceed for the clinical, but still we are more into preclinical investigations. And now we our collaborator from US have collected produce amount of scientific evidence on this. They keep on producing that, and maybe in few years it is ready for clinical trials, clinical investigations. So the important ongoing public health debate concerning the balance of potential health benefits and safety against potential adverse effect or toxicity of Kratom and Kratom products calls for improved oversight of regulation of the US Kratom product market, as illustrated by a series of the recent articles in the Tampa Bay Times published in December 2023. So if you go for the Tampa Bay Times, you can see that there are a series of articles being published on the potential health benefits, the issues of safety concerns, adverse effect toxicity of the Kratom and Kratom-related products. The authors uncovered hundreds of fatalities attributed to consumption of products sold in the United States and vast, complicated intricacies of the currently unregulated US trade in product claimed to be derived from the Kratom plant. So there's a lot of issues going on in US whereby the Kratom product itself, they uncovered some of the fatalities attributed for the consumption of the product, being sold in US. So there are several comprehensive reviews of kratom alkaloid chemistry and pharmacology have been recently published. So if you go through and search about that, there's a lot of information recently been published about this. Interestingly, the issue that we face in the West, does not occur in Southeast Asia region, especially in Malaysia and Thailand, because there's no fatalities or adverse effects were reported in this part of the region till now. So whatever been reported in the West, especially in US, it doesn't occur in the Southeast Asia. There is something that the scientists from that part of the world, they are wondering why in Southeast Asia, there's no such thing happen with this consumption of this plant. So these are some of the images of the product being sold in the market, especially in US. So you can see that there's a lot of labeling, name, Borneo Kratom. So if they say about Borneo Kratom, is mostly is come from Indonesia part of Borneo. And you can see they have a Malay Kratom, Malay Kratom, which is attributed to Malaysia, but and also Thai, Indo, Maeng. So they have these names, You can see also in these products, red barley, green barley. So there is claims also these kratums are being cultivated or produced from these regions, Red Borneo, green Malay. So green, red, white means they try to attribute it with the veins of the kratom leaves. So neither kratom or any of its active components are listed in the schedules of the United Nations Drug Conventions, UNODC. So a lot of you are aware that kratom is a psychoactive plant, that gives psychoactive effects. It especially is alkaloids, So so its active components, which is particularly referring to the alkaloids, they are not either of them are listed in the schedule of the United Nations Drug Conventions, but the United Nations Office on Drugs and Crimes had urged all the member states to stay informed about the current developments and trends related to the kratom and their respective countries and regions. In Southeast Asia, countries including Malaysia, Myanmar, and Singapore list kratom and its active component metagenin and 7-hydroxymetagenin, which is a oxidative derivative of the metagenin, under their controlled drugs law, and these two compounds are classified as a Class A controlled substance and are governed by the Misuse of Drug Act, MDA. Especially in Malaysia, the use of kratom is prohibited and strictly regulated in the Poison Act 1952, with the penalties for possession and trade, So, of course, the botanic name, which I mentioned just now, Metagenes speciosa, in Malaysia known as ketom or biak-biak. In Indonesia, if correct, is known as kadamba, puri, or keton. And in Myanmar, it called as binsa, binsayawat, which is a tree. It is indigenous to the Southeast Asia region itself. The plant is frequently cultivated in the northern region of Peninsular Malaysia and throughout Thailand, mostly in the southern part of the Thailand and in West Kalimantan province of the Indonesia on the Borneo Island. So the locals from southern Thailand and northern Malaysian Peninsula traditionally consume the aqueous decoction of freshly harvested leaf. So if I'm highlighting here because it's quite important, it is quite different from what reported from the West, where they're consuming a processed leaf products, in terms of extracts, dried leaf powders, and so on. But here, we are consuming, traditionally been used as aqueous decoction, decoction of the freshly harvested leaf for its distinctive medicinal properties to treat a variety of ailments such as diarrhea, muscle pain, and hypertension. So metagenin, which is later I will elaborate on its chemistry, it is the prominent alkaloid found in the leaf. In the class of alkaloid, it can be primarily isolated from the leaves of this medicinal plant, Metagenes speciosa cod. So the first isolation of metagenin, was 100 years ago, it's over 100 years ago by Alan Field, 1921, and later confirmed by Josie et al. And Zacharias using early spectroscopic techniques. So Ellen Field thought that she isolated mitragenin, but then it's only get to be confirmed in the year of 1963 and 1964, yeah, the structure of the mitragenin itself. So earlier studies, they only mainly focus on mitragenin as congener, but actual mitragenin content in crude extract was not clearly defined. So Ponglux, yeah, in the year of 1994, reported that 66% mitragenin in the young Thai leaf and 12% in the mature Malaysian leaf, though based on the limited samples. Yeah, so it is a clear, it is reported. Yeah, but this 66%, 12%, it is reflecting to the total alkaloid content derived from the extract. Yeah. So recent study shows a wide variation in mitragenin levels across region. So generally, we found out that it is 10% or less, not as what been claimed by these reports. Yeah, indicating earlier values were likely overestimated. So the variation in mitragenin content could be attributed to several factors that intrinsically affect the main constituent composition, such as maybe due to the different chemotypes, climate, environmental pressures, soil type, as well as the type of extraction method and solvent use. Yeah. So basically, when they say that in the leaf, it is actually referring to their total alkaloid content, but generally it is 10% below. So I will give you some example on that. Yeah. So this is a study conducted by my student, and previously we can see that different type of extraction method been used, which is chocolate hot maceration, cold maceration, ultrasonic assisted extraction, as well as this new extraction technique, which is accelerated solvent extraction. So basically from this, we can see that the mean percentage of the mitragenin itself, we can see that within the time, though we can see that variation of time itself, chocolate using chocolate, hot maceration, cold maceration, they're using more time compared to the other two technique, which is just in minutes we can produce that. But we can see basically correspond to the time. We can see the extraction, the mean percentage of mitragenin, though it looks similar, but you have to attribute to the time used. So these two techniques, they produce mitragenin content which is in the lesser time. So mitragenin yield is extraction dependent with solvent polarity and method determining the efficiency and resulting content. So it is one of the proofs. Yeah, we can see that it depending on the extraction type. Yeah, and as well as the other extraction variables such as time itself. So here within the same technique, which is UAE and ASC, we can see that by using organic solvents such as methanol, ethanol, and ethyl acetate, we can see there's no significant difference observed, and it shows a quite considerable amount. Yeah, it is can say that also we can see that it is below 10%. So, but it's consistent between the organic solvent compared to the aqueous. We can see the aqueous is very less. So mitragenin is actually is a semipolar alkaloid with the lithophilic properties. So it explains why it has higher solubility and extraction efficiency in organic solvents compared to water. So the question here is when we see the traditional application, people are using aqueous decoction. So in aqueous decoction, the issue here is what is the mitragenin level in there? Because current research is heavily focused on mitragenin. But then when we go into the traditional preparation, which is the aqueous decoction, what is the level of mitragenin in that? So because of the mitragenin's properties, which is semipolar alkaloid and lithophilic properties, yeah. Now we are entering the chemistry of kratom and its phytoconstituents. Okay. So apart from mitragenin, the other interesting alkaloids are also present in a considerable amount in M. speciosa, which is kratom, especially in the leaves. Yeah. To date, about 55 alkaloids has been successfully isolated and identified from these species, which begin from the isolation of indole alkaloid mitragenin. Yeah. Subsequently, other alkaloids from the leaves were discovered. It has its diastereomers, yeah, mitragenin isomers, pisugenin, pisusilatin, and mitracilidin, were some of the prominent indole alkaloids that found in the leaf. Corresponding to the mitagenin, the content of analogs of mitagenin also varies significantly based on the regional varieties and the maturity of the plant. For instance, the leaf of the matured and speciosa plant contain the main alkaloid. When I talk about the plant, it is actually referring to the leaf. So the main alkaloid mitagenin is the diastereomers, which is phytogenin, phytosilatin, as well as its congener penantin, yeah. While the leaf of the younger plants contain mitagenin, speciogenin, phytosilatin, and a small amount of the mitagenin diastereomer, which is mitrasilatin. So mitagenin having its diastereomer, 3 diastereomer. Penantin is congener. It is actually an analog of mitagenin, yeah. So chemistry, again, yeah, although this plant has been widely consumed due to the medicinal properties such as analgesic effect, mitagenin have been found to be the major alkaloid in the traditional herb despite having opium-like effect at higher doses, which is reported by Wilson, yeah. The local agriculture community commonly consume it as a stimulant to increase endurance and counteract fatigue while working under the hot sun. So people those days are using it as a to counteract fatigue, yeah, fatigue, increase endurance, yeah, like energy booster and so on. So several studies indicate that mitagenin exhibits similar effect as cocaine, yeah, coca-like effect also, and opium on the body. So this is what they report. So you can see that its phytoconstituents, you can see that it's mainly, though there's a phenolics, flavonoids, yeah, phenolics, flavonoids also include in phenolics, but there's phenolic acids and flavonoids, but the kratom plant leaf is actually heavily focused on these two types of alkaloids, which is mitagenin, which is the indole alkaloid, you can see from the structure, and it's congeners, yeah, because they're having the similar backbone structure. And another one is the oxyindole. So this is the indole alkaloids, and this is oxyindole, which is found in the kratom plant, yeah, especially in the leaves. So when we call them as mitagenin congeners, because having the same backbone structure, which is the tetracyclic, yeah, we can see that it is highlighted in the yellow, which I explain later. And also the difference is just because of these substituent groups, which is substituents and configuration, yeah, that makes them different from its parent structure, which is mitagenin. So we can see the example of the indole alkaloids, and these are the oxyindole alkaloids, which is reported from the plant itself, yeah. Now we are entering into the chemistry of mitagenin, which is the prominent alkaloid. So we can see that the mitagenin is a prevalent monotropic indole alkaloid that is found in the kratom with the molecular formula of C23H30N2O4. So this is the structure of mitagenin. So we can see that this is the indole part, yeah. Mitagenin, also known as a 9-methoxycorinantidine, has the distinctive characteristic of solubility in alcohol, chloroform, acetic acid, and it is partial solubility in water. So mitagenin also have a similar characteristic to Uncaria and yohimbine type alkaloid. So Uncaria and yohimbine type alkaloid, which is characterized by its indole structure composed of bicyclic structure of phenyl group fused into 5-member pyrrole ring. This one, yeah. And also 10-carbon corinantidine type structure derived from the terpene biosynthesis. Okay, so referring to the tetracyclic structure. Furthermore, mitagenin is biosynthesized from the tryptamine, yeah, derived from amino acid tryptophan, and cycloganin derived from terpene biosynthesis before forming cytosidine and undergoing a series of intramolecular reaction to form. Yeah, this is relevant to the biosynthesis, yeah, to form mitagenin. Moreover, the indole portion of mitagenin was substituted with the methoxy group, yeah, OCH3 group on the C9 here. This is C9, this is the methoxy group, yeah, on of an aromatic benzene ring, whereas the quinolenzine ring comprises of corinantine type carbon framework being attached to the carbomethoxy and olefin methoxy group, yeah. So this is briefly on the biosynthesis of mitagenin. So we can see that it is from the shikimic pathway as well as the NEP pathway. And This is the compounds, yeah. So the indole structure been derived from this pathway. So we can see the indole part and as well as the terpenoid part. So combined to become the monoterpenoid, yeah, indole alkaloid. And from the cyctosidins forms, undergo series of the reaction and undergo to become mitrajanin. So in the recent publication, yeah, by Scott, in the year published recently, 2023, they have elucidated on the directed biosynthesis of mitragenous stereoisomer. So we can see that when they report on the mitragenous stereoisomer, so it begins as what I showed before, is cyctosidin, but it undergoes of the series of the reaction to form mitragenin. And it is from the corinantidine. But this corinantidine have two types here. 5A refers to 20S, 5B refers to 20R. So this is the 20S and 20R here. But only 20S corinantidine may able to produce mitragenin. Whereas if there is an inversion of configuration happens here, it could form speciogenin, which is 20R. So the configuration itself is very important in producing the right alkaloid, yeah. So structurally, mitragenin has three chiral, which is asymmetric center as C3, C15, and C20. So C3 here, C20, and C15, yeah, C15 here, you can see that. So respectively, which gave rise to stereoisomers, speciocelatin, speciogenin, and mitracelatin. It agonistic activity also depends on the structural activity relationship, which directly correlates to the three major functional sites that is prominent for opioid binding receptor, which is the phenolic hydroxyl here, and then benzene residue and tertiary hydrogen. 7-hydroxymitragenin portray a high binding affinity towards opioid agonist receptor. So about this, I will explain later, yeah. But 7-hydroxy already, the OH is here in this B, yeah. So towards opioid agonist receptor being compared to the morphine and mitragenin. So from the SRS studies, there are 4 factors that determine its activity, which is the existence of the substituent group on C7. C7 is here, and C9. C9 is here. A and B, yeah. Modification of beta methoxyacrylic group and the nitrogen lone pair. So beta methoxyacrylic group. So we can see that what happened is this modification happens in the each sites here. This is the sites of the compound. So here, beside methoxy, if you substitute the long alkyl chain, so OH, it lead to the decrease in activity. Whereas in B, presence of OH group increases the activity. So when attachment of OH here, that what makes it 7-hydroxy mitragenin. Whereas in the C, the N-oxidation abolishes the activity. D, the double bond decrease the activity. So the double bond here, you can see is a congener penantine, yeah, having a double bond here. And R configuration abolishes activity as well in E, yeah, here. And then NH3 replacement abolishes activity at the F side here. And then G here, ester reduction decreases activity. So any modification have to take account of these aspects. You know, if you want to increase the potential activity of particular activity you desire, such as pain or anti-cancer activity, you need to consider these sites, which is very important for its pharmacological applications. So about the chemistry of mitragenin and 7-hydroxymitragenin. So 7-hydroxymitragenin recently is heavily debated. So majority of Kratom research to date has focused on the chemistry and pharmacological of mitragenin, along with its oxidative derivatives, which is 7-hydroxymitragenin. Why I'm calling it oxidative? Because you know, the part of the OH attached to here. And it is reported to be less than 2% of the content, whose pharmacological effects remain conflicted and under debate. So this 7-hydroxy is heavily debated, and I will show you about the report where it is been reported, yeah, recently, a recent report on that. So mitragenin, so this is mitragenin, the parent compound, and its oxidative derivative, which is because of the OH attached here. Yeah, only one substituent group. It makes a big change. So mitragenin is a prevalent chemical constituent in the plant. It is used as a chemical marker. Yeah. So whenever we assess, even in the for enforcement and regulatory agent, they use mitragenin as a chemical marker to detect Kratom products. Good solubility, it has a good solubility in alcohols and other organic solvent, but poor solubility in water. Yeah. But then when 7-hydroxymetagenin, yeah, which is an oxidative analog or metabolite of metagenin, which having an extra OH in the structure, it is the minor constituent and the existence of this compound still debatable. Whether naturally occurring or post-harvest derivative still is being debated, yeah, whether this compound is naturally produced inside the plant itself or it is maybe due to the post-harvest, it can be a post-harvest derivative, post-harvest effect, yeah, which, metagenin get oxidized, yeah, to form 7-hydroxymetagenin. Good solubility in alcohols and other organic solvents, but it also has a better solubility, improved solubility in water than metagenin, yeah, because of this OH itself. So this is the issues related to 7-hydroxymetagenin. So it is in the recent report by US FDA, yeah, 7-hydroxymetagenin, an assessment of the scientific data and toxicological concerns around an emerging opioid threat. So you can see that the 7-hydroxymetagenin itself is a big issue. Yeah, it's been heavily debated. So in the executive summary, you can read here that if you're interested, you can find this report to get to know about this 7-hydroxymetagenin. Yeah. So the recent report indicated the increased availability and marketing of 7-hydroxymetagenin in the US, raising a public health concern due to its pharmacology. So critically, yeah, I just highlighted several few important points to highlight here, but you can go through if you have time on this report. Critically, 7-hydroxy produces respiratory depression, physical dependence, and withdrawal symptoms characteristic of classical opioids such as morphine, fentanyl, oxycodone, and hydrocodone. So just by adding our OH here in the structure of metagenin, yeah, it can produce such effects. Yeah, this is reported in the FDA report. But then they still stating that it's a naturally occurring substance, though it's been debated. Yeah. And then it's concerning proliferation of the concentrated 7-hydroxy product that are sold over the counter and online, which means products labeled as Kratom, they've been concentrated with 7-hydroxy. Remember when I mentioned that 7-hydroxy, yes, so in this information, yeah, you can see that 7-hydroxy comprising less than 2% of the total alcohol content is very less than 2%, yeah. Even some reported, yeah, 7-hydroxy content by the weight range 0.01 to 0.04%, and some even less than 0.05%, yeah. And also here they're mentioning about, yeah, this is a recent report, yeah, Sharma Idia 2025, where they reported on the less than 0.005%, yeah, to a maximum of 0.21%, yeah. So you can see that the content of 7-hydroxy is very less. How come suddenly there's a concerning proliferation of the concentrated 7-hydroxy products? They are actually from the natural substance. I mean, naturally is from the plant itself or is being synthesized. Yeah. So this is the concerns. Yeah. So you can see that the 7-hydroxy in this product are likely synthetically derived to oxidate chemical conversion of metagenin isolates. Yeah. Metagenin. Yeah. For metagenin or kratom extracts. So these are the examples that we can find in internet, yeah, about the 7-hydroxy product. You can see that, yeah, they have different flavors, yeah, it's a chewable tablet, yeah, some of these product, yeah, they have been sold, yeah, in the Western markets, yeah, especially in US. So recently, yeah, the American Council of Kratom Consumers, they come up with these 10 points, 10 reasons why the natural kratom leaf should not be grouped with the isolated concentrated synthetic product as a public health concern. So this is important for me because it's a major issue concern in there. So this is why it become interest to study this plant, because there's a lot of things that we can get to know, a lot of things that issues that a lot of research being conducted in chemistry, pharmacology, clinical, preclinicals, and so on. So one of the reasons we can see here is actually, number one, the FDA recommended scheduling concentrated synthetic 7-hydroxy, not natural kratom leaf. Here itself, we can see that, they are differentiating the 7-hydroxymitragenin from a natural kratom leaf. And because of the potential risk, yeah. And then you can see that the second point they are mentioning about the centuries of traditional use show natural kratom leaf is safe, especially when they said about these indigenous people from generations when botanists arrived in Southeast Asia in the early 1800, yeah. So they can see that natural kratom leaf is safe because of the, when I mentioned earlier, you remember where people use it, they chew the leaf or they use primarily using aqueous decoction. And then some of the important key here is number 5, mitragenin does not cause respiratory depression like concentrated synthetic 7-hydroxy. So there's a distinctive difference between mitragenin and 7-hydroxymitragenin effects. Yeah, though there's just the difference is just because of this OH at the position carbon 7 and so on. Yeah, so these are the things that they highlighted about this. This is the recent issues, yeah, they're ongoing on the debate on the plant itself. So this is about the, if you see from this FDA report, one of my research are being cited, yeah, they suggest 7-hydroxy present botanical kratom relatively low or trans amount, maybe because of the post-harvest oxidative derivative of mitragenin. So this is the research that I reported, whereby we conduct a chemical composition of water ethanol liquid extract preferred from the freshly harvested leaf from Malaysian Kratom. So freshly harvested means we have a good amount of time, which is within 2 hours we harvest the leaf and we prepare the water and ethanol extract, the fresh leaf. Yeah, we did a study on the fresh leaf. So we, as I said, yeah, most of the published research on the kratom to date, they focus heavily on the chemical and pharmacological property of mitragenin and 7-hydroxymitragenin. So in Southeast Asia, freshly collected kratom leaf or the water-based preparation, which is the decoction, are consumed. So when we talk about the leaf and the chemical constituent is always about the phytochemicals. Of course, in the leaf, they contain a lot, multiple phytochemicals. So in our study, we use aqueous and ethanol extract, liquid extracts, yeah, prepared using kratom leaf, freshly collected in the northern Malaysia, and we analyze freshly harvested, yeah, in northern Malaysia, and we analyze using a validated UPLC ESI QTOF MSMS at the Center for Drug Research, Universiti Sains Malaysia. So this is the data. It's very interesting, yeah. When we do the MSMS analysis, we identify, yeah, the presence of 7-hydroxy in ethanol extract compared to water. In water, we identify the presence of 7-hydroxy in water extract, not in the ethanol extract. So this is an interesting finding, yeah. You can see that I highlighted here in the 7-hydroxy. The 7-hydroxy compound number 3 is only found in water extract compared to ethanol. So this raises a question: why is present in water and not in ethanol? So basically, when you use an organic solvent which is quite stable, yeah, we didn't found the presence of 7-hydroxy, whereas in water extract, because we use maceration. So macerations, of course, it takes days, yeah. So when we use maceration method in water, we know that it promotes oxidation, yeah. It's a good oxidation agent. So it might, convert mitragenin into 7-hydroxymitragenin, whereas we didn't see that, we didn't observe in ethanol extract. So 7-hydroxymitragenin were identified only in the aqueous solution. So yeah. So this is the findings, and this is the publication that we report on that, yeah, about the one of the key highlight is the presence of 7-hydroxy. So of course, when we see another thing is it is very subjective to the analytical instrument or spectroscopic method they use, because when it comes in detection, because 7-hydroxy reported only a traceable amount, but most of the analysis using LC-MS. How about NMR and other analytical techniques? So we have to go and look into and compare it whether between these analytical techniques, whereas there is a distinctive observations on the presence of 7-hydroxys there or not, yeah? So the chemistry of metagenin and its diastereomers. Metagenin congeners, especially diastereomer, were found to be present, yeah, in the leaves, which I mentioned, speciogynin, speciosilatin, and metacillin. So these are the structures of the compound itself. And since diastereomic phytoconstituents are congeners of metagenin that have a tetracyclic indole alkaloid structure, this compound can be distinguished through the structural configuration at certain important positions. So basically, the position are the C3, C15, and C20, which result the occurrence of the metagenin and its diastereomers. So the summary of the absolute configuration and positioning and respective compound is shows here in the table 1. Yeah, so you can see the metagenin C3. Yeah, C3 here. This is C3, C20, C20, and here C15. So this is the three stereoisomeric center here. Yeah. So you can see that the configuration, the metagenin, speciogenin, speciosilatin, and metacillin. C3, yeah, as configuration, whereas for speciosilatin, metacillin, they have RR. And for C15, it is always the same. Yeah? All of them are in S configuration. Whereas in C20, there is difference between metagenin, which is S, speciogenin R. So that's what makes that metagenin differs from speciogenin. And then as configuration and R. So here, which make differs from metagenin as well as speciosilatin and metacillin. So these are very important, and it is hardly can be distinguishing just by using mass spectrometry because they're having the same mass. So the techniques that can be used is NMR, as well as if you use LC, yeah, in combination with LC, liquid chromatography, where specific retention time, you can observe the elution of these compounds. So again, yeah, the example of metagenin congeners, the structure. So yellow represent the same core structure. So from yellow, you can see metagenin. So you observe the tetracyclic, yeah, bone of the structure. So it occurs in all of these compounds. Yeah, most of them having tetracyclic, which make them to be a metagenin congeners, because metagenin is a predominant alkaloid in leaves. Blue indicating different functional groups. So you can see the different functional group. As I mentioned, penantine, you can see the vinyl and vinyl moiety here, as well as this red, yeah, the OH group. So basically, isomers, diastereomers, congeners, despite having a very similar chemical composition, they are likely to have different chemical and pharmacological profiles, yeah. So this is another interesting issue that I found out. So basically from the HPLC DAD analysis. So this is metagenin. Yeah, A is metagenin, B is penantine. Penantine is this one. So we can see metagenin and penantine is difference in C20. One having ethyl group, one having the vinyl, yeah, the double bond. So both of them, they are congeners for each other, yeah. So even the case of congener, you can see that. And then 7-hydroxy. So this is 7-hydroxy. We can see the elutions of this compound, basically from the retention term, 7-hydroxy eluted first, followed by penantine and meta. Of course, this is a method dependent. It depends, but basically in the method that we develop, so we able to see of the compound. If you analyze separately, you can see the chromatogram of each of the compound. But then what happens if you having a mixture? So you can see a problem occurs here, whereby 7-hydroxy, we have no problem. You can clearly distinguish that. But what happened to the metagenin and penantine? They eluted together. So this is one of the challenges if you use analytical methods such as HPLC, yeah, to determine the metagenin and penantine. So this is one of the issues that you need to be aware, and clearly using a comprehensive advanced detailed method, analytical method, in order you can separate them well, because in most cases they have been found together and it is can be merely observed in HPLC. You need MS, when you observe in MS, then you can get to know, because they are congeners, they're having different mass. One is one having different mass from other. So 398 and 396. So you can see that when you observe in MS, then you can see that occurrence of the two different masses, then you know it's a mixture, it's not a single compound. Yeah. So if your positive ionization method, of course, yeah, 399, 397. So if you use MS in a positive ionization, you can observe, yeah, the occurrence of the compound. So the importance of the diastereomers and the congeners, you can see that these corinantine type indole alkaloids, they are inducing opioid agonistic activity. Yeah, opioid agonistic activity. There are some reports by Takayama et al. 2002. And also mitagenin pseudoindole, and 7-hydroxy. Yeah, 7-hydroxy mitagenin were found to act as opioid agonists with higher potency than morphine, which is reported by Takayama himself. Yeah, Takayama and group. And then another report, yeah, mitagenin is analog, yeah, penantine isomer, speciogenin and speciosilatin were found to act as opioid agonists, which found the leaf extract of M. speciosa exhibit opioid agonist effect. And then 7-hydroxy mitagenin showed the most potent opioid effect. So you can see that several reports, you know, when they observe and see the activity of these mitagenin and its congeners as well as and its isomers, yeah. So future direction of the Kratom research and development. So what are the future direction of Kratom research and development? So a lot of people are debating heavily on the issues of the Kratom. Of course, we can see that there are more chemical profiles of this plant being established, but then there's still a gap issues that need to be solved. But then when we're talking about research and development, where are we now? Where are we heading towards? Are we having sufficient scientific evidence and findings, especially in terms of chemistry, pharmacology, yeah, safety profiles, toxicology, and so on? Is Kratom ready for the next phase, especially clinical phase? Nutraceutical product, are we into that? Or botanical drug development? So these are the questions, the things that we need to think about, since you know, there's a lot of scientific evidence being produced, and we need to think that where Kratom will head next in the future applications. So this is a bit on the published in Journal of Natural Product. If you're talking about botanical drug development, yeah, US FDA perspective, we see on the scientific and regulatory approach. Over 800 botanical investigational new drug applicants, yeah, and pre-in meeting requests. From the 800, you know, we only, you know, they only allow, yeah, permit 2 botanical new drug application. I mean, they approve in the US, yeah, in the year of 2006 and 2012. So it's a bit challenging, you know, to obtain a approved botanical drug, yeah. So we have the guideline for the botanical drug development for the industry by the US FDA. So one of the key important, we must have the quality control by chemical tests, sample analytic tests such as spectroscopy or chromatography method that capture the active or chemical constituent of the botanical drug substance, and manufacturing control process validation. So here, it is important to establish a proper chemical profile. Analytical means proper method. Yeah, we need to have that, you know, the chemical profile of the Kratom itself need to be established well. And then botanical drugs are generally heterogeneous mixtures. So of course, we are aware of that. And as such, their chemical constituents are not well defined. So these are the challenges, you know, when we investigate botanical drugs, yeah, herbal extracts, you know, they often are mixture, they are complex mixture. So in some cases, the active constituents are not identified and their biological activities are not well characterized. And the case of isomers, you know, the analogs, this is pretty much that we, especially isomers, yeah. So in the quality control test, you can see that, you know, quality description of the drug substance, it should include the name, appearance, and active constituents. So you can see that it is important. So chemical, for the quality control test, chemical identification for the active constituent, if known, and the chemical constituents. So in general, the sponsor should use the best available analytical technology to address the issue of analytical resolution. When resolution is inadequate in one particular method, multiple methods should be used to provide complementary data for adequate chemical identification and quantification. Also, quantification of active constituents, if known, or chemical constituents. If several botanical raw materials are combined to produce a multi-plant substance, a quantitative determination of each individual active or chemical constituent is infeasible. A joint determination can be made for several active or chemical constituents. When multiple active or chemical constituents are known, they should be chemically characterized and their relative amount of should be defined. So here we can see that it is important, yeah, our analytical and spectroscopic method. So in conclusion, yeah, kratom-related issues should be handled carefully with proper scientific measures and analytical technique to produce sufficient scientific evidence that will assist in kratom botanical drug development. A comprehensive and detailed chemical techniques need to be carried out in order to be fully characterized and identify the kratom's phytoconstituents and its content as regulated by the relevant authorities, yeah, by US FDA and PRA, of course, yeah, and in each country we have our own regulatory agencies, yeah, based on the provided guidelines for botanical drug development and pharmaceutical applications. Proper and sufficient chemical information on kratom, whether it's in dry material, extract formulation, or constituents, will lead to a more reliable and precise kratom botanical drug development. So thank you. This is my research group in my center. Yeah. So all of them combining this multidisciplinary. So we have a pharmacist, neuroscientist, pharmacologist, social science people. They'll be working together in addressing the issue related to kratom and addiction. Yeah, and as well as pain. So this is my beautiful research groups. Yeah, my students. So they contributed to my research. Yeah. And then, yes, thank you very much for listening and willing to allow me to present on this topic. Yeah, thank you very much. So this is my contact. So anyone who would like to collaborate with me, you are welcome. We can discuss about that. Thank you. So I pass to Prof. Rafi. Prof. Rafi is here.
Maybe Prof. Raf still on the lecture.
Oh yeah, he's on lecture. Okay.
Yeah.
So if anyone want to discuss, I can discuss about the topic. Yeah.
Sorry, Dr. Chiru, can I ask you some questions?
Yeah, sure, no problem.
Yeah. First of all, allow me to introduce myself. Is Pinus. I'm from Department of Pharmacy, Universitas Islam Indonesia. And I think we have met before when I visited Center of Drug Research at USM with Dr. Ansmi.
Oh, okay. Yeah. Yeah. That's why I saw your name and I was thinking that it's a familiar name that I might meet you. Yeah. How are you, doctor?
I'm fine. I'm fine. Thank you. And it's really nice to meet you again. And thank you for your such a nice presentation about kratom, which is kratom in Indonesia still also has a issue with the authority and about the legality, something like that. Maybe it's the same as in Malaysia. Thank you again from the chemistry of kratom, which is quite interesting. I have maybe 2 or 3 questions if you don't mind. First about the mitragynine, which is the concentration of the mitragynine in the leaf influenced by when the leaf is harvested, or also the states of the leaves. I mean, like the mitragynine concentration is the highest percentage, the content in the leaves. I mean, like you know that the leaf is there's maybe the young leaves, or that's so when the time for harvesting. And then secondly, about the hydroxy derivative of mitragynine, you mentioned about which is the minor component. you said that it is still debatable this is naturally occurring or the post-harvesting. I'm just wondering if it is post-harvesting, does it mean that the hydroxy derivative of mitragynine was produced automatically when the leaf is picked? So when the leaf is picked, so the situation is already different, right? It is because I have an analogy with this, like because I work on sponges, which is the sponge we take from the sea and then outside from the sea. So the oxidation it's occur usually. We cannot avoid that. So is it because of maybe automatically produced by after pick or harvest, or is it because during the process when from the pick up and then after that there's sortation and then what do we call it, the storage, something like that. So that's my questions. Thank you very much.
Okay, thank you, Dr. Pinos. Actually, the first question, I think I almost forget. I think you are mentioning about this harvesting of the leaf, right? At the stage, what stage we should harvest, something like that, is it?
Yeah, this is about the in which stage or what time or what when we pick or harvest the leaves. I mean, is it maybe after like 3 months or that's after or the young leaf that we need, something like that?
Okay, actually, to answer that, there's still research going on that, which is in Thailand as well in Malaysia, because most of our source sample lives actually from the plantation, and we don't know what is actually their method of practice. Yeah, because either they're plucking the leaves actually from the young trees or the old trees, or old means mature trees, or uh, maybe in different which part of the nodes of the part of the leaves as well. But then of course this is something that need to be pointed out, because we are not clearly knows that whether the leaf used to produce the products are whether from which stage of the plant itself. So to answer that, we currently in the stage of research, and we are still doing the research whether which part of the leaf produce a specific. But there's some research being produced in US actually. But in US, I think if there's a paper on that, which they're mentioning about different type of these kratoms, and which type of the chemical constituents that are there. The content itself varies. But then in our part, we still in the midst of research and determining on which period will be the how to say that the period that is suitable for us to plug the tree for the application part. Yeah, but then of course it cannot be young, because there is a distinctive chemical evidence with the young and old. There's chemical variations that we can see in terms of the phytochemical content. So that is something we need to investigate on then, and we're still in the midst of investigation. Second is about the 7-hydroxy. The 7-hydroxy is heavily debated, and we can see from the report, some they put it there as a naturally occurring. But then if you see in some of the paper being produced in Thailand, and we also have some evidence that we will produce later part, which is this 7-hydroxy, but using HPLC, HPLC analytical method, we didn't observe that. But then we did observe when we analyzed the abstract, we observe in LCMS. As we know, LCMS is very sensitive technique. So when people reported that 7-hydroxy is present, it might be in a traceable amount. LCMS the mass able to detect that. But whether it's a naturally occurring or not is debatable, where I believe a comprehensive and extensive research need to be conducted. But 7-hydroxy, from what we found out, that is the 7-hydroxy might be, this is what we hypothesized, might be the post-harvest derivative occurs from the conversion from mitragenin to 7-hydroxy mitragenin. But it present in the leaf part in a traceable amount. It's not as much as what people are reported.
Okay.
Yeah.
Yeah. So
But whatever in the market, as what I show in the report, it has been synthetically produced.
all right,
Yeah.
thank you very much. There is another question, I think, from another participant.
Yeah.
I think there's many question in chat, Doctor. Maybe can you have a look?
Okay.
Yeah, I saw that there's question there.
Okay. So where we start, yeah? There's a lot of questions. Okay, let me see. Okay, so there are some asked about this Kratom to determine, help analyze Kratom. Oh, okay. So in CDR, we do have a lab that is established in order to analyze Kratom, yeah, Kratom analysis, specifically mitragenin. So you can, if you really want to perform the analysis, you can contact our center. We can help you with that. Yeah. But then if to Indonesia and Malaysia, I don't know, because we have different law governs, right? Indonesia, I'm not sure about the law. It's not certain, right? But in Malaysia, it's highly regulated under Poison Act. So we need to obtain a permission from in our part, we need to obtain the permission from NPRA, National Pharmaceutical Regulatory Agency. So we need to obtain and need to declare, then only we can proceed with analyzing this compound and this extract. Other than that, any other questions? Or you can ask here maybe. Oh, okay, dari Dr. Handy, yeah. Without mitragenin, speciogenin, penantin, 3-isopenantin have the same molecular. Actually, mitragenin, speciogenin, yes, they have same molecular formula because they are isomer to each other. Penantin and 3-isopenantin, they are isomer to each other. So to analyze them would be difficult because having a same molecular mass, it is hardly can be distinguished in mass spectrometry. But of course, with the method development in liquid chromatography, we able to see the distinctive separation of this compound. But then without a standard, yes, it is hard to identify because, as I said, it depends on the instrument and method analysis, method development. So each method development can result in a different separation and different RT. Yeah. So in terms, we can annotate, but of course, if you use the same method again, we can annotate. But if you develop a different instrument, you might need standards to validate the position where it eluted. Okay. Yeah.
I think there is someone, Ibu Nur Sarafana raise her hand, I think.
Sharafana, yeah, my former student actually.
Oh,
Yes.
oh no, okay. Please, please.
Okay. Hi, Dr. Tiru. Long time no see.
Hi.
So
Nice.
my question is really simple because as a phytochemist who has done so many work in this field, I was just wondering, in terms of phytochemical standardization, what kind of challenge that still remain before a kratom-based formulation can meet the quality and also the reproducibility standards?
Okay, so that is a very good questions from a phytochemist. Yeah, for phytochemist point of view and also analytical chemist, yeah, because kratom having a complex mixture of phytochemicals, especially when you see the alkaloids. Alkaloids are very well known to have diverse isomers, yeah, within themselves they have isomers and the stability issues. Of course, the challenges will be to establish a full comprehensive analytical chemical profile. So of course, in order to establish that, we need to develop a method, a fully established method in order to separate, distinguish them each and them in a and clearly can be distinguishable. But then the issues that we're having here, especially in analytical analysis, especially HPLC, because of course it's heavily dependent on what type of instrument we use, type of mood. So the result can be varies. So even in NMR itself, when you go for metabolomics, you produce the profile, it is in a complex mixture. So in a complex mixture, you hardly to this, even though you can distinguish isomers in NMR, but then distinguishing congeners itself is quite how to say, challenge, because mostly in NMR we can see a specific region for alkaloid. All the alkaloids like mitragynine congeners, they will accumulate there. And then it's very hard to distinguish whether this is mitragynine, speciogenine, or speciosilatine. You understand? So you need a standards. You need the standards to develop an overlay to see where they've been positioned. Yeah.
I see. All right, thank you for that answer, Dr. Thiru.
Okay.
Oh, it's nice to see. Okay, my name is Rudy, Doctor. I'm also from the Department of Chemistry.
Okay.
So maybe I have a question actually. Yeah. So yes, as Dr. Pino said that in Indonesia actually we don't prohibit it or don't legalize the use of kratom.
Yes.
So and I think the major issue is like that. Suppose that the kratom is can give like addiction. Yeah. So I do what because and actually I wonder is so in Indonesia we have three type of we call the botanical drugs. The first one is jamu, and the second one is standardized extract, and then the third is the phytopharmaca. So the difference is the minor difference is the category is depend on the depth of the scientific evidence, right? So I wonder if I think jamu usually we can use daily without any strong evidence for the pharmacological effect. But then because kratom is, I think, in Indonesia also people can traditionally use. I think maybe the same as in Malaysia. So, what is your opinion? Is it based on your Phytochemistry? Is it possible that maybe we can use kratom for our jamu if we just process by like aqueous extraction?
Okay.
So in compare with the toxicity or addiction effect. So what do you think, Doctor?
So, I mean, I cannot answer for the global, but I can answer from my perspective and what we believe, actually, our team of research in Malaysia, but I represent my opinion myself. Actually, if you ask my opinion, Doctor, I believe that kratom should use for medical purpose as what as so if you use for nutraceutical or other application, I think it's hard to be regulated or controlled because it can be abused, right? If you use as a botanical drug, which is administered under the Ministry of Health, then we can regulate and we can make sure that the usage of the drug itself is for the therapeutic use, especially for pain and addiction treatment. So it can be safely regulated by the agencies, and then it can be used by people who really need, because when we talk about drug itself, it be used whenever it's necessary to be used for treatment. So that is what our stand is. Actually, we want kratom to be a drug that been used to treat specific diseases. with the permissions, yeah, from the health ministries in our country lah. So
Okay.
it is, if we want to use as a nutraceutical, I'm afraid that it might having challenges in terms of regulating or enforcement, because it can be misused if
Mhm.
is in a mere markets as compared to if you market it as a drug which is monitored and regulated by the government agencies. So that is my opinion. I think you might agree on that.
Okay, okay, okay.
Because I
Thank you.
think that is the regulation itself, the abuse, the issues that we're facing, why we had to get a green light to use
Mhm.
this, because of this abuse and the abuse potential,
Uh-huh.
the issues on the regulatory, how we regulate that, yeah, something like that.
Okay. So why is different with the weed?
Again?
Like our neighboring country, like Thailand, is actually I don't know. I still free for using the weed.
Oh.
So what is the difference? Like, actually, maybe the concerns between the pharmacological effect between the weed and the kratom. What do you think?
I think it's quite different. I think, if not mistaken, because there are different pathways, right? Kratom is more,
Okay.
yeah. So that's why. I mean, I think they are reconsidering to how to say, tarik balik. Because I mean,
Yeah.
always have issues on regulatory, and the potential to be abused is always there, right? So yeah, that is the issues that always
Yeah.
yeah, and how to manage that, yeah.
Okay. Thank you, doctor, for your explanation. And
Sure, no
is
problem.
there is there anyone want to ask to Dr. Suventan?
But I think Indonesia also through BRIN, yeah, they're exploring Kratom, right? They're doing
Yeah.
Kratom research. Actually, recently I went to Bali. We did Kratom updates, and I can see researchers from Indonesia is also venturing, exploring the science of Kratom inside their research about Kratom, because most of the Kratom products, yeah, the source
Mhm.
is in Western markets are reported to be coming from Indonesia. It's not
Yeah. I think that one is mostly coming from the Borneo, yeah, from Kalimantan.
Kalimantan, yeah.
Yeah, from Kalimantan. Yeah.
So yeah,
I read from the information that actually kratom is only from export, but I'm not sure if it can be used in Indonesia as a kind of
yeah, I don't think so. I think it's for export, if not mistaken as well. Yeah.
yeah. I
I
don't
mean,
know if for the consumption in Indonesia, I think it's not like
traditionally people do practice that. I mean,
yes, yeah,
but in terms of the product itself, we don't have a clear, right? Clear.
yeah, yeah.
Yeah, I think still I think BPOM Indonesian FDA still prohibited to use the kratom a part of the our botanical drug. So
So Indonesian FDA is considering that.
yeah,
But it can use, but if in a regulated approach, I think it shouldn't be a
Mm.
problem, because
uh,
for drug to be used under a regulated approach, I think shouldn't
Mm.
be a problem, because it's controlled, right? Controlled drug.
yeah.
Yeah.
So I have one question, is you don't mind?
Mhm.
Yeah.
Sure.
Yeah, as we know, kind of another alkaloid like a morphine is I think it's addictive, and it has the derivative of morphine is dextromethorphan. So
Mhm.
I'm just wondering, is there any derivative synthetically modified mitragynine that can make the derivative less addictive?
If less additive, I'm not sure about that. But whenever people try to modify, they make it more, they increase their activity, right? Rather than
Yeah.
reduce the activity. But then
Because
I think there's some maybe there's some are doing into that, medicinal chemists maybe looking into that, while maintaining is potential pharmacological activity,
yeah,
but reducing it. But the issue is they are binding the receptor binding itself,
yeah.
because opioid receptors, right?
Yeah,
Yeah.
yeah,
So,
because the dextromethorphan actually is kind of has a similar like morphine with
mhm.
used as in the like is for the cough medicine or something like that, but it's less addictive. But I heard that actually it's already misused sometimes because of the still there's a addictive.
Yeah, yeah,
Yeah, I'm
true.
just
I mean,
wondering if Dragonin has the derivative that make less addictive, but the activity is the same or something like that.
so far from now, we didn't heard about that, but we only know compound that is more potent than mitragynine itself, like 7-hydroxy. And this 7-hydroxy itself been synthetically produced, and the product is marketed in
Mm.
markets as shown just now. So this is what FDA heavily debated about it, and they're worried because just by adding one OH in mitragynine, it becomes 7-hydroxy mitragynine, which is highly potent, yeah. It exhibit agonistic behavior compared to
Ah,
mitragynine itself, the parent compound. So when people trying to market the product itself, it raises a debate and concern on that, because I think for 7-hydroxy, because it's a limited amount in plant, so it is hard to correlate. But when we have a sufficient amount of mitragynine, I mean 7-hydroxy mitragynine, and we do preclinical investigation, then we can have a series of data on that can prove. But now still until now, the results are still debatable and not conclusive yet. So we still waiting for our groups here. We currently doing research on that. Even our international collaborators, they are heavily doing research on that to make sure that clearly distinguishable between kratom leaf extract, mitragynine, 7-hydroxy mitragynine. This is the focus that they are currently focused on. I think Indonesia is also doing good job on that. They are starting to explore, right? The potential applications. Yeah.
so Pak Rudi is becoming the host.
So I think Prof. Rafi is still in lecture, is it?
Yeah,
Okay,
probably.
it can be, yeah.
Oh, okay.
Pak Rudi becoming host.
Usually, the speaker will close the the meeting
Yeah.
by themselves.
I I mean, if they have any questions, or we can still have some time, I think. If not, then
Yeah.
you are free to discuss. You can email me. I mean, I've showed my email, and you can contact me.
So there's
Yeah.
one comment, doctor. This one that I think from Pak Emil, yeah, said that the standard is expensive. So do your group actually already like isolate the mitragynine as a standard, or
Yeah, we isolate compounds as and 7-hydroxy as well. But then 7-hydroxy is very less, so that's why it's very expensive. Yeah,
okay.
that's why it's very less. It's a lot of procedures, and it's very
Uh,
less, so it become more
uh,
expensive compared to mitragynine itself.
okay, okay. So I think uh, so if someone from Indonesia willing to collaborate with your group, so it means that we do extraction in Indonesia, and then because we don't have any standard, then can we like try to send the sample? Is there any regulation to send the sample to Malaysia?
In Malaysia, we do have
Or,
regulatory body that governs that. We need to ask permission for that. I mean, from our side and from your side as well, because it is heavily regulated drugs, right? Even
uh,
when
uh,
the extract contain mitragynine, we need to declare. But then, if for research purpose, yes, we can have a collaboration between the institute and we
Mhm.
ask them permission to do research. I think they can permit us to do that.
Okay, okay,
But
okay.
we have to declare. We have to declare the amount, the things that we need to do. We need outline the research properly, and then they will give us but they will come and audit us for that as well.
Okay, okay.
But it can be done. Yeah, it can be done. You can
Okay, okay.
help on that. Yeah, sure.
Oke, thank you. Masih ada yang mau ditanyakan ke peserta yang lain?
Saya boleh tanya lagi nggak?
Silakan Pak, waktunya kan masih ada 9 menit lagi.
Okay. Dr. Thiru, so in the Center of Drug Research at the USM,
Okay.
it's not only focusing on the kratom, right? I mean, like if there's other what is the other plants or other compound that has a potentially as a candidate for drugs, would it be possible to have a collaboration, for example, to exploring the potential of some compounds for drugs, something like that?
Yeah, sure. We welcome that, especially is when it comes to psychoactive, because our center's niche is addiction, and
Oh,
we exploring to addiction and also in some in pain. So
hmm.
this is our main focus. So if anything related to that, we are welcome that to do a collaborative effort on research. Yeah.
Oh, okay. So it's addictive and the pain.
Yes.
Okay.
Pain and additive.
Oh.
And this is quite interesting because I mean we are fully focused on Kratom because our partners collaborators from US, they actually want to having a quick fast data, and then we all focusing on Kratom now to because it's if you can see in recent 5 years there's a lot of publication coming out in Kratom, and either in pharmacology, some preclinical evidence, also in chemistry itself. So our main focus here to make sure that the research, the fundamental aspect of the research is completed, and we move to the botanical drug development. Yeah.
Thank you, Dr. Thiru.
Yeah. You're welcome to collaborate, Doctor.
Yeah, thank you. Hopefully I can visit.
Silakan.
Yeah, Ken, please come. We do have MOU, right? In
Yeah,
USF.
we have MOU.
Yeah, please come.
Oke. Masih ada yang mau ditanyakan sedikit lagi, mungkin satu pertanyaan? Oh. Okay. I think there's no question anymore, Doctor. Yeah, I think we can close our
Yes.
webinar today. I think thank you for your kindness to sharing research about Kratom. Maybe later we can do more collaboration with Indonesian part for about research in Kratom. Thank you very much, Doctor.
Thank you for inviting me. Thank you. And
Yeah.
please send my regards to Prof. Rafi as well. Thank
Okay. Okay.
you. Bye.
Bye bye.